Hello guys, today we'll be talking about the biochemistry. The important most topic is glycolysis, which we have learned since our 12th standard. So let us go into this topic in detail. What is this glycolysis actually? Glycolysis, breakdown of glucose. So it is a major pathway for oxidation of glucose and produces the energy than HMP pathway, which is the minor pathway of glucose oxidation. Now let us talk about this glycolysis in detail. So this glycolysis is also called the EMP. EMP means the embedded Meyerhoff pathway. Embedded Meyerhoff pathway. Now, what is a site? So, where does this glycolysis occur? It occurs in almost every cell in the cytoplasm. Okay, and yeah, it occurs in every part of the body, in every tissue, every cell, and the site of occurrence is cytoplasm. Now, this glycolysis is a 10-step pathway. So I have mentioned here it's a pathway, not a cycle. So this cannot be regenerated. So it's a pathway like this, it's a straight thing, not a circular thing. Okay, so this is a 10-step pathway. Now let us look into each step in detail and know how the energy is liberated from the glucose. Okay, yep. Now the first thing is, first step, glucose when enter into any cell get first phosphorylated into glucose 6 phosphate. Okay, now this reaction is catalyzed by the enzymes hexokinase or glucokinase. Hexokinase, it is present in almost every cell, but glucokinase is mostly confined to the liver. Both of them perform the same function like converting glucose to glucose 6 phosphate. So when as phosphate is added here, now what is the source of phosphate? The source of phosphate here is ATP. ADP it has become after donating one phosphorus. So as here phosphorus is being added, so this reaction becomes a phosphorylation reaction. So we told now this glycolysis is a main pathway for oxidation of the glucose and produces energy. Now see, in the first reaction itself it have used up the energy. Now what was the thing, then what about the thing what we have told, like it produces the energy? See, for anything to produce, first they require an investment, just like a business. See, only when you put an investment you finally gonna get a profit. So in the similar way, this glycolysis also, it first requires energy in the form of ATP, later even it will be producing energy in the form of ATP. Okay, so this glycolysis I can tell you in three phases. So one is the investing phase, one next is the splitting phase, and latter is a producing phase. Okay, the investment, splitting, and then producing phase. Now the first step itself has utilized the energy in the form of ATP, the phosphorylation step, and the enzyme catalyzed here is hexokinase or the glucokinase. Now coming to the second reaction, the glucose 6 phosphate has converted into the fructose 6 phosphate. We know glucose, fructose are isomers. As there is a change among the isomers, the enzyme catalyzed here becomes isomerase. And this is a reversible reaction if you observe. And this reaction is isomerization reaction. Now coming to the third step. The fructose 6 phosphate now get converted into fructose 1,6 bisphosphate. So here one more phosphate is added. So again same, the source of phosphate comes like ATP. Now it has become ADP. Now what is an enzyme? So whenever a phosphate is added, of course the enzyme could be the kinase. What kinase here? Phosphofructokinase 1. Phosphofructokinase 1. This enzyme is very important. This is the rate limiting enzyme, and this step is the rate limiting step in the entire glycolysis. What is this rate limiting step? It is a committing step. That is, if this step has occurred, this after this everything has to occur. Okay, so that is why it is called the rate limiting step. If this step has occurred, there is a surety that this glycolysis is definitely going to happen. That is what this rate limiting step mean, and this is the rate limiting enzyme. And now as phosphorus is added, this comes again, this reaction is a phosphorylation reaction. Okay, now later, so the third step has completed. Now coming to the fourth step. So what happened in the fourth step is a cleavage. I've told you till now we have invested. Now the splitting has to take place. So the splitting occurs and it becomes G3P, glyceraldehyde 3 phosphate, and DHAP, dihydroxyacetone phosphate. Dihydroxyacetone phosphate. This fructose 1,6 bisphosphate will be having 6 carbons. As it has splitted, it will be having 3 carbons. It will be having 3 carbons. Okay, here the enzyme mediated this splitting reaction is aldolase A. This is a bond cleavage reaction. It's a cleavage reaction. Now what is the fifth step? Fifth step is DHAP is converted into the G3P, glyceraldehyde 3 phosphate. This reaction happens with the help of the enzyme mutase. So this reaction is keto aldo isomerization. Keto-aldo isomerization. Now, how many molecules of G3P is formed? By aldolase enzyme, one molecule is formed. By this mutase enzyme, another molecule. So, finally, after the fourth step, how many molecules of G3P we are having? We have 2 moles of G3P. Now, please do remember this 2 moles of G3P because all the energetics depends upon there. And if any question has been arised, they also has to be thought in the point of view that we have got 2G3P. Okay, remember the point 2G3P. Now, yeah, this 2 moles of G3P, glyceraldehyde 3 phosphate, now it has to produce energy. So it has to produce energy in the sense its molecule has to become a stronger enough, a stable enough. Now what happened is we will be adding a phosphate here. Now what happened? Glyceraldehyde 3 phosphate, it becomes 1,3 bisphosphoglyceraldehyde. Bisphosphoglyceraldehyde. Now according to our theory, so whenever the phosphate has been added, where should it has to come from? It has to come from ATP. But no, this is not happening. So this is a special reaction. So this step number is 6. So the sixth reaction, the phosphorus has been come from the cytoplasm. It's a very special step, and here the phosphate is added without utilizing ATP. This is a speciality of this reaction. How the phosphate is added? By using NAD. So as here 2 moles we have, so 2 moles of NAD plus 2 phosphate ions, it becomes 2 moles of NADH. So this 2 moles of NADH will enter the ETC, electron transport pathway, and produces 5 ATP. So we know one NADH will be producing 2.5 ATP, and therefore 2 moles of it has produced 5 ATP in this step. Okay, but this happens in the aerobic respiration. Okay, this happens. Do remember, the formed 2 moles of NADH in the 6th step will produce 5 ATP via electron transport pathway only in the aerobic respiration, not in the anaerobic. I'll tell you what happens in the anaerobic later. Yeah, now the formed 1,3-bis-phosphoglyceraldehyde. So it is also a 3 carbon molecule. It is also having 2 moles. We have the 2 moles. Now the molecule has become big enough. Now we have to produce energy. So in order to produce energy, we have to break the bond. So we'll have to break. So how do we break? So we'll remove a phosphate. So if you remove the phosphate, what happens? 2 moles of ADP has accepted the removed 2 moles of phosphate and become 2 moles of ATP. So here we have formed ATP. Okay, here this reaction is mediated by the enzyme phosphoglycerokinase. See again kinase, the removal of phosphate is also taking the final ending kinase. So this reaction, the seventh reaction where there is a production of ATP, this is the substrate level phosphorylation. Okay, substrate level phosphorylation. Now what the product has formed? So the phosphate from the first position we have removed. Now the 3-phosphoglyceraldehyde. 3-phosphoglyceraldehyde. So again, do not forget 2 moles. So this 3-phosphoglyceraldehyde. Now the phosphate present in the second position is not that strong enough, that stable enough. Now we have to convert we have to change the position of the phosphate. Now let us make this reaction PG mutase, that is phosphoglyceromutase. By the action of this enzyme, it has converted into 2-phosphoglycerate. 2-phosphoglycerate, again 2 moles, don't forget that. 2-phosphoglycerate. This reaction, this is the eighth reaction. This is an isomerization step. Okay, so this 2 moles of phosphoglycerate, it undergoes next reaction comes like by the action of enolase, it forms phosphoenol pyruvate. 2 moles of phosphoenol pyruvate, PEP we usually call. This is this reaction, this is a ninth one. It is a enol bond formation. So you can easily remember this enzyme enolase. Phosphoenol is formed, so enolase we use. And now coming to the next reaction, the final 10th step. This 2 moles of phosphoenol pyruvate. Now let us completely break this. We'll break this enol bond. Now what happened? This enzyme, we know breaking of this bond which we require. Pyruvate kinase. So this has broken the bond in this way that enol has gone. Phosphate is removed in the form of ATP. See again, 2 moles of ADP has become 2 moles of ATP. Now finally what has left over? Pyruvate. A 3 carbon molecule of 2 moles has finally formed the pyruvate in this glycolysis. Now glycolysis has not stopped with this pyruvate. Now whenever there is a conditions of the anaerobic, so anaerobic conditions when takes place, what happens? This pyruvate will be converted into lactate by the enzyme lactate dehydrogenase. So in this reaction, what will happen is 2 moles of NADH has become 2 moles of NAD. Okay, so this happens under the anaerobic condition. So whenever the lactate is formed, there will be nothing else will be formed. That is called the dead end of glycolysis. Okay, now from where this 2 moles of NADH has come? We have told you now in this sixth step where oxidation and phosphorylation is happening, 2 moles of NADH under the aerobic condition goes and produces 5 ATP. So under the anaerobic condition, what happens is the 2 moles of NADH will come in this reaction and will form the lactate, and therefore in that step 5 ATP will not be produced. So that's why I've told you that reaction happens only in the aerobic conditions. Okay guys, this is all about the steps. And now coming to the things to be known in this glycolysis. Now how many irreversible reactions happens? The first one, hexokinase reaction. Hexokinase, phosphofructokinase, and pyruvate kinase. So literally to say, almost all the kinases are irreversible reaction except the phosphoglycerokinase, which is an irreversible reaction. The only kinase which is reversible is PG kinase, that is phosphoglycerokinase. Now how many steps you find the substrate level phosphorylation? 2 steps. One is the phosphoglycerokinase and the pyruvate kinase, where 2 moles, totally 4 moles of ATP has been produced. Okay, these are the 2 SLP reactions, and these are the 3 irreversible